it's Raph. Good evening, good morning, and good afternoon to all of you. So, for tonight, I haven't been streaming in a long time due to a lot of stuff in college. We're doing a lot of interesting labs and a lot of just craziness, but fun craziness from college and all of my classes. And it's now actually test season, so a couple of tests are coming my way actually next week. So, why not study for these upcoming tests? So let's do a little bit of like just a chill stream here. We're just going to go through some of the first chapter assignment problems, study guide, whatnots for the test, and then hopefully chapter 2 and chapter 3 and so forth and so forth. All right, let's get started. Thanks so much for everyone who's joining in, and without further ado, we'll also have some music in the background. This will be Symphony in D Major, Opus 2, First Movement from Vincent Graffard. So let's play some background music here. Let's get started. Alright, so which of these statements is consistent with the data presented in the evolutionary tree if the trait of photosynthesis did not arise in the fungi branch? Right. So Saccharomyces, that's yeast. Drosophila is a fly. I forgot about Canarbedillus elegans. I forgot what that was. I think that's a plant. Okay, let's see. So since yeast is a fungi, and it's talking about the trait of photosynthesis did not arise in the fungi branch of that tree, I'm going to go with A. We're gonna have to delete that B though, so I'm gonna just hit backspace. There we go. Alright. Which is the following statement true or false, and why? Which is not evidence for the endosymbiotic origin of mitochondria and chloroplasts. So there's this theorem basically that's saying our mitochondria and the chloroplasts of plants and algae were free-living proteobacteria long time ago that got engulfed by a larger cellular organism. So let's see one of these options which is going to be taking that into account. Let's see. Contain their own DNA, which is not evidence for the symbiotic theorem. I think this one, because since there are two different species of bacteria, they don't have similar DNA to each other. They do have bacterial DNA, so got that one correct. Alright, scientists were studying yeast, and a mutation has arisen and activated a gene required for division. Without this gene, the protein it encoded, the mutant yeasts were unable to divide normally. You could rescue the mutant yeast cells, allowing the cells to resume normal division by adding a human protein. So based on this finding, what is the most likely conclusion you could make? So let's see. Okay, yeast and humans diverge from a common ancestor. I do not think that's correct, because our common ancestor with yeast is an extremely distant one. Yeast cells require human proteins to divide? No, not really, because normally they didn't need the human protein, so that one's incorrect as well. Let's see, yeast cells most likely obtain their cell division proteins from the cells of early humans. I don't think that's correct either, because I think fungi evolved, much earlier than humans did, obviously. D. Proteins that control cell division in yeast and humans are functionally equivalent and have been conserved almost unchanged for more than 1 billion years. I think that might be a possibility. Let's see. E. Modern humans most likely obtain their cell division proteins. Yeah, I don't think that one. This one kind of sounds like pseudoscience, and we don't like pseudoscience. Let's go with the scientific sounding one here. The proteins are conserved over time. Which sounds... I think that this one might be correct. Let's see. Alright. Which statement is not true of chloroplasts? Each has an internal stack of membranes and is enclosed by 2 membranes. Alright, so we know this one is actually true, so it's not the answer, because In chloroplasts, you need to increase that surface area. You need to find a way to make sure all of that's going good for all the reactions. You need a lot of surface area in your chloroplasts. So that one's not going to be it. Let's see. What other options? Chloroplasts absorb light and generate oxygen and carbohydrate. Or chloroplasts contain DNA. We also have chloroplasts, or that's in the symbiotic theory, so that one's probably true. Present in essentially. Okay, so all eukaryotic cells, that's a red flag. We don't have chloroplasts, even though I think it would be pretty amazing if we did have chloroplasts, we would be able to make our own energy. So I'm gonna hit that one because it's basically asking which one is false. I'm gonna hit that. Alrighty. Okay, let's answer. Alright, we did it. Look at that. Translation occurs in the... So translation is the process in which mRNA is converted into your proteins. The mRNA travels out of the nucleus into the matrix of the cell in the cytoplasm. So I think that's the answer, but it needs to meet a ribosome in order for this protein to be made. So let's put cytoplasm. Alright, we got it. What is the name of the process by which eukaryotic cells engulf material captured from an external medium? Okay, so endocytosis, not cytokinesis. I know for sure it's not cytokinesis because that's cell splitting. Endomitosis, that's weird. I've never heard of that term, so I don't think that's that. Endosymbiosis, nope. That's talking about like the endosymbiont theory. Again, mitochondria were originally free-living bacteria that were engulfed, but I don't think it's talking about that. Let's go with endocytosis. Let's see if this one's the lucky guess. Alright, I think we got it. Hello, Adventures of Anna. How are you? Hope everything's going well. Thank you so much for tuning in. No, I can't skip class. I have an 8 a.m. class tomorrow. It's a lab. We're looking at fruit flies. I can't skip that. Yeah, I can't skip that class. It's so interesting. We're looking at fruit flies, and we're building, like, these simulations in Minecraft, too. I don't want my professor to be mad at me, either. Alright, which statement is not true of mitochondria? I think we just saw this one, too. So, mitochondria are thought to have originated from bacteria. That one is actually true because of the endosymbiont theory. They're actually engulfed by bacteria very early on in the history of life. Mitochondria are involved in the chemical energy cycle. That's true because they're the powerhouse of the cell. Mitochondria have an inner and outer membrane, so this one is true as well because they were engulfed. So they still had their original membrane from when they were bacteria, and they were just engulfed by a larger cell, so that one's true. Mitochondria contain their own DNA. That one's true as well because they are originally bacteria, and what's something that's really interesting is they're actually studying the DNA sequences of mitochondria, and they're trying to find like evolutionary changes and mutations in that DNA sequence of the mitochondria, which is pretty interesting. So E has to be the one that's false. So let's select E. Just gonna copy it over. And there we go. Alright, we got it. Genetic changes followed by selection are best described as the fundamentals of what process? I think, so selection, that's like Darwin. So I'm thinking evolution. Just gonna type it. It's my favorite topic since, in Pokemon there's a ton of evolution that goes on. So, let's evolve it. Let's not be like Ash Ketchum. Ash Ketchum, never evolves his Pokemon. He is the world champion, though, so maybe there's some merit to it, but He doesn't really evolve his Pokemon. They're just like strong off the bat. So hats off to Ash Ketchum. Alright, evolution. There we go. We got it. Alrighty. Which organelle's ancestor was likely engulfed by primitive eukaryotes to help the cell survive in an oxygen-rich atmosphere? Alright, so I think it's the mitochondria, so that'll be D. Again, mitochondria is like plural, and then mitochondrion is single, so there we go. Outwards to inwards, the structures of the cell under an electron microscope would be outer membrane, cell wall, plasma membrane, and then, so we're out of the membranes now. We're just inside the cell. So I think the best answer is cytoplasm. There we go. The genome of E. coli contains 4.6 million nucleotide pairs, times 10 to the 6 nucleotide pairs, whereas the human genome contains some 3,200 times 10 to the 6 nucleotide pairs. So the E. coli has more nucleotide pairs, which means its sequence is longer, basically. What could be concluded based on these numbers? So E. coli are unicellular, whereas humans aren't. That is true, but you can't conclude that based off just the genome. You need to do some other tests or other analyses for that. Human cells are larger. You can't really conclude that based on the numbers either, because it's talking about the genome. Human cells have 700 times more genes. No, that might not be incorrect because you're just looking at the genome, so you can't really tell that either. Humans are a more complex life form. Yes, that's correct, but you can't tell that from the genome because it's just the genome. So we know E is not true because not all of them were correct, so F has to be the correct answer. None of the above conclusions were drawn correctly. There we go. Which structure could not be seen using the electron microscope? Alright. So DNA. So again, the electron microscope, it's like the most powerful type of microscope we have available to the public, I guess. If you want to play like a little conspiracy theory thing, I guess you could say like, oh, the electron microscope's the most powerful one they have in the public. But in reality, you have like the TEM and the SEM, but those are just types of the electron microscope. You could see things like large macromolecules, stuff like that. You can't see, I think you could see atoms in some of the more powerful ones they have nowadays, but the electron is actually a fraction. It's a very minuscule fraction of the mass of an atom. So I'm gonna guess C. You can't see electrons using the electron microscope. Kind of self-explanatory a little bit, in a way. There we go, we got it. What is a drawback to using electron microscopy? Okay, so it requires the use of fluorescent probes. Nope, that's fluorescent microscopy. Can it be used to view living cells? That's kind of true. Can it be used to view a whole cell or an organism? That's also true, I think. Atomic details. Structure is larger than a ribosome. Cannot be used to view. I'm going to go with this one because you need to code it and stuff. I guess you could still see a whole cell or an organism if you wanted to, because there's a lot of tiny organisms, but I'm gonna guess living cells, because if I remember correctly, the light microscope, the dissecting microscope, and then one of the fluorescent microscopes are the only things that could see living cells, so electron can't, so let's hit that one. Alright, we got it. Is the following statement true, false, or impossible to determine? One thing all cells have in common is an ability to colonize any environment on Earth. So this is very tricky because if we're talking about any cell, this will be false because you have things like archaea. So basically, what's the correct word for this? Very hardy and resistant microorganisms called archaea, and they actually live in some of the most extreme environments on the planet they could live in very salty regions. They could live in very hot or very cold areas where usually you won't have any signs of life, but archaea can be found here. You could find them in places that don't have oxygen. You could find them in sulfur pits, all these dangerous and crazy deadly areas that you would think, nope, life can't exist here. But you could find archaea, and they are these single-celled, very resilient microorganisms. So you would think false because they could actually go there. But it does say all cells, so it's talking about all types of cells, so not just archaea. So that means I think I got the answers mixed up, but I'm actually going to say that this is false because all cells can't colonize everywhere, but some can colonize in these more extreme conditions, so therefore it must be false. Oops, I hit the keyboard button instead. My bad. There we are. Which of these cannot be resolved with a conventional light microscope? Alright, so light microscopes, you could see single cells, but you can't see tinier things such as the ribosome, for example. You can't see the mitochondria. You kind of can, but you need to be very, very good at trying to find it. A ribosome will be on the surface of like the endoplasmic reticulum, and it's extremely small, so I'm going to go with a ribosome because it's basically a protein with 2 subunits. Alrighty, we got that one. Which statement represents the cell theory? Alright, all cells resemble square or rectangular chambers. I don't think that's it. All cells contain DNA. That may be true. All cells require continual input of energy to sustain life. That one's very true. All cells can be seen using a microscope. That's also true, but that sounds like the odd one out. All cells are formed by the growth and division of existing cells. Okay, cell theory. So it's not talking about the foundations of living things. So those other answers were probably foundations of living things. The thing that is cell theory, you have to have cells in order to make other cells. So let's go with E. I think the music's kind of getting a little bit loud, isn't it? They must have had a crescendo written in this part. This is an original piece by Mr. Vincent Giffard. So it sounds like an actual like romantic era symphony, so it's pretty cool. Shout out to Mr. Vincent Giffard for writing this wonderful piece of music. You just turn it down because the crescendo is kind of picking up on the monitor there. Alright, we got that one correct. When comparing liver cells and kidney cells within an organism, many differences can be observed and documented. Which of the following is not a difference between liver and kidney cells? The different cells express different genes, maybe. Different cells produce different proteins. The different cells are generated during the animal's development. The different cells have different roles in the body, possibly. I'm gonna go with D, but it also sounds like it might be B or A. I'm not really sure on this one. Let's just see. Oh, study this one. So we got one incorrect. They have different roles. Did not see that one. Let's see. Different DNA. Okay. Alrighty, we could just make a note of this one, so we need to study this one so they have different DNA. Whoopsie daisy. Alrighty. An electron microscope is used best to show which object? Chemical bond. Ooh. Which part of the cell different proteins congregate? No, that's going to be fluorescent. Mitochondria are very small. You could probably use a chemical bond. I didn't know about that one. Let's do mitochondria. It's either mitochondria or chemical bond. I really think it's mitochondria because you could look at a single one. Alrighty. Is the following statement true, false, or impossible? At a molecular level, the members of the two domains of prokaryotes, the archaea and the bacteria, differ as much from each other as either does from the eukaryotes. And this is true because they wouldn't be in They wouldn't be in two different groups, basically, if they're so similar, the taxonomy of things. Archaea and bacteria are extremely different from each other. Why do cell biologists study yeast? Yeast is a model organism. It's a eukaryote, but it's also a single cell organism. So you could just look at the processes of a singular cell. We're also eukaryotes, so they have like the membrane-bound organelles that we do. And they're easy to grow, and also they have a lot of other uses too, like baking, you know. Yeast carries out all the basic tasks. So I think it's that one. It's number A, or letter A, I should say. A is not a number. Alrighty. Let's copy that in. There we go. Alright, we did that one. Oh, we're supposed to choose a few more of them. Which is not a function of proteins. Casing certain viruses, encoding genetic information, providing cells with structural support, catalyzing biochemical reactions, or acting as molecular motors. Encoding genetic information. I think it's that one. Because these two sound like enzymatic processes, but uncoding, oh wait, ribosomes translate it, but they don't encode it, so I'm taking a gander at B. Alrighty, we got it. Which cellular component separates the DNA of eukaryotic cells from the cytoplasm? It is the nuclear membrane, or the nuclear envelope. It's a little package that keeps the nucleus safe inside the cell. It's its own little, like, compartment, basically. It's a little room. Alright. There we go. 1970, Fry and Eden published research describing the mobility of plasma membrane proteins. They fused mouse and human cells together. They made a hybrid cell and then examined the localization of mouse and human proteins over time. And eventually they moved over the entire cell instead of just being half and half. And what technique did these two scientists use to examine the mouse and human proteins? So we're looking at proteins. So we have interference, light microscopy, probably not that, transmission, scanning. We're not looking at the protein itself, nor are we looking at like a very detailed surface in a small scale. I think it's fluorescence, because then you can mark certain proteins against other proteins in other parts of the cell. Then you could see that they migrated. So I'm going to go fluorescence. Ooh, don't want to copy all of that. We just want a tiny bit of D. There we go. Alrighty, we did it. Alrighty, let's see this next one. A model organism that is microscopic, single-celled, and eukaryotic is... Then we have a bunch of scientific names. I know B was this was a plant, and then E. coli, again, bacteria, so prokaryote. That Saccharomyces is our yeast, our friends the yeast. They're eukaryotic and single-celled, so I'm going to pick D. Little tiny fungi, that's what they are. Alrighty. Which of the following is a stack of flattened membrane-enclosed sacs that receives molecules made in the ER, often chemically modifies them, and then directs them to the exterior of the cell or to various locations inside the cell? Okay, so this could only be one thing, but let's go through our options. Peroxisome, that's a lysosome that will get rid of peroxide. So it's basically like an enzyme that gets rid of those bad, nasty hydrogen peroxides inside the cell. So it's like cellular waste. So it's not that, because those don't do any shipping and handling. They're not Amazon Prime. Lysosome, these are enzymes that secrete different chemicals to get rid of cells, because lys means to basically kill or end a cell. So lysosomes do that job. When there's a bad cell, just kills it by acting on it, and so you got that function there. ER, endoplasmic reticulum. I don't think it's that because that's where lipids are made. So, nope. Ribosome, that's where the protein is made from mRNA. It's not about shipping or handling there. They're not, again, Amazon Prime. So the Golgi apparatus, there's our Amazon Prime. There we go. Free shipping. There's our shipping center in our cell. There we go. Which is the smallest object that can be seen using an electron microscope? Okay, so we can't see an electron. We could see an atom, I think. Could probably see an atom. It's either individual molecule or atom. I'm going to go to atom because I've seen pictures of atoms from microscope slides and stuff. Oh, study this one. Hydrogen atom, okay. Did I not? Oh, individual molecule. Alright, so that must have been just like... Well, it could have been just like a smaller molecule. Those images were pretty misleading on this one. Alright, let's just continue. What organisms commonly colonize the hot acid volcanic springs, the airless depths of marine sediments, the sludge of sewage treatment plants, the pools beneath the frozen surface of Antarctica, as well as the acidic, oxygen-free environment of a cow's stomach where they break down ingested cellulose and generate methane gas. Alright, so these are all like extreme environments. These are basically the Ohio of ecosystems and habitats for life. So there's only one group of organisms metal enough to survive in these crazy environments, and that has to be the archaea. There we go. Alright, so transcription is DNA to RNA. We haven't left the nucleus, so it's still going to be in the nucleus. Which of the following is a model plant used by scientists? Alright, so our elegans. So it's that C option. Homo sapiens, we aren't plants. I don't think so. Plants are people too, but people aren't plants. So it has to be the plant one. This one. Really? I guess not. I was studying this one wrong. Don't know my scientific names. So it's actually that Thaliania one. I need to see what this elegans thing was. Let's see. I'm gonna look it off Google off stream. Let's see what this elegans is. I'm gonna look really stupid brain when we see what this thing is. Oh, it's a roundworm, a nematode worm. We were thinking a worm was a plant this whole time because I didn't memorize my scientific names. That is very dummy of me. Alright, let's see what this one is. Is the following statement true, false, or impossible to determine? Like the differentiated cells in an individual plant or animal, all bacteria have the same DNA. Nope, because you have different species and therefore they'll have different DNA. So, I have to copy this whole answer. We can't just type in false because they'll actually mess with the system here. There we go. Within the developed multicellular organism, all cells possess the ability to divide and do so regularly. Okay, not all cells divide. Some cells, yes. Because you have things like the skin cells on the outside, the thing, calluses, that's actually dead skin cells, so they can't divide anymore. It is false because there are some cells that lose this ability. There we go. Thank you, Anatomy and Physiology 1 class. What do eukaryotic cells have that prokaryotes lack? So, big deal with us, eukaryotic brothers and sisters and otherwise. We have a nucleus and membrane-bound organelles that our prokaryote friends don't. So, let's pick that. Alrighty. Mitochondria are essentially the same in all eukaryotes, including plants, animals, and fungi. Based on this observation, how were mitochondria most likely acquired? So this is again that endosymbiont theory, where they are engulfed by a bacterium, and they used to be just like free-floating, free-living bacterium. They're like proteo. I looked up the species one time. I forgot what it was. I should have wrote it down. I've always had the question, like, what type of bacteria, like, what species exactly were mitochondria? I looked it up. I found it. I forgot to record it somewhere. But anywho, what's your options? By an ancestral prokaryote, then lost in line? Nope, that's not true. That's not true. B is not true either, because 1,000 years ago, there are people even further back, and even eukaryotes, much further back than just 1,000 years ago, so I don't think it's that. By an ancestral eukaryotic cell, then replaced by chloroplasts? No, I don't think so. By ancestral eukaryotic cell before the lines that led to animal cells, plants, and fungi diverge. Let's see. From a free-living photosynthetic bacterium. I think it's that one. Let's do C. It's either C or E. I'm betting on C. We could also put both of them and then just hit, I was correct. I think there's an option if we're really 50-50ing it. Okay, so that one was 50-50, so we're just going to override it. Is the following statement true, false, or impossible? An individual ribosome can be seen with the fluorescence microscope. It can be marked, but you can't see an individual one. You'll have a lot of like green specks if you mark ribosomes green. You can't zoom in on this fluorescence microscope, so an individual one cannot be seen, so false. Oh, looks like I need to study. This one I'm not going to override because this one was my bad. You can see a ribosome even though you can't see an individual one. You could still see ribosomes. I was kind of, batting around the bush there. Whoopsie. Alright, what is a drawback to using light microscopy? It cannot be used to view structures smaller than a bacterium. It cannot be used to view living cells. That one is false. Requires the use fluorescent probes. That's incorrect. We're not using fluorescence microscopy. Cannot be used. It could only be used. Cannot be used to view structures smaller than a bacterium. I'm gonna pick A. Alright, fingers crossed. There we go. I got an 85% on that one. That's not that bad. Very good score on it, I guess. Oh, is it going back again to... What happened here? These are like the ones we've already seen, so I'm just gonna start copying them over again. I think it just like repeats them after a bit. Okay, so these are the ones we missed. So, liver and kidney cells. So they, which of the following is not a difference? So they produce the same proteins, express the same genes, or don't. Which of the following is not a difference? Let's see. So which ones are false, basically? I think it's the different cells have different DNA because genes, sequence. It's all about sequence. So DNA. There we go. Which of the following is a model plant? We just went on in the tangent for this one. It's the Arabidopsis thaliana. However you say it. Apologies if I butcher that name. Following statement true, false, or impossible to determine. We just saw this one as well. A ribosome can be seen, even though you can't really zoom in on it on the fluorescent microscope. On the electron microscope, you could really see a single ribosome really well. Did it not work? We could have just typed it in, but this is faster. It's supposed to be faster. There we go. What is the smallest object that can be seen using an electron microscope? Okay, I think this one was the hydrogen atom. You could see a singular, or was it molecule? Oh yeah, hydrogen atoms are too small. You could see small molecules, you just can't see the building blocks of them. It's not that powerful, or at least the ones during when the book or wherever the questions were from are made. Okay, let's do this. There we go. 86%, not bad. Alrighty, I think we're going to do like round 2 and stuff tomorrow, so let's just call it a day here, this study session. Thanks so much, everyone, for tuning in. Thanks so much, Adventures of Anna, for also joining me. I always appreciate when you join. Thanks so much for tuning in. Hope everything's going well with you as well. Alright, thank you so much, everyone, for viewing this stream and, I guess, learning, picking up some of the cell bio stuff as well. Yeah, please have a good rest of the week, and also please do something nice for someone. Good night, everyone, and thank you again.